Can you hear me if I stand? Good, thank you. So, thank you for the introduction. My name is Antonio Collesei, and I'm a researcher in bioinformatics at the Veneto Institute of Oncology in Italy. The work I'm going to talk about today is called SHISMA, which is an algorithm that is able to infer significant subnetworks from time series in single-cell data, and that are shape-driven and cell-type specific. Allow me to start this talk with a little joke. In fact, they say that in the landscape of single-cell algorithms, there are only two trends that are mainly relevant. That is de novo reconstruction of gene regulatory networks and trajectory cell lineage inference. And we are going in another direction. We are like with this other stuff. But as someone very famous and very controversial would say, let's make other stuff great again. So let's jump on it. So what we're talking about, as I said, is subnetworks discovery from time series data. So these are the keywords. So let's introduce a little bit about time series analysis, but not too much because I'm pretty sure you know about time series. This is a time series. It's about average temperatures essentially in the last 2 centuries. And as you see, it's really long. I mean, it's taken like continuously. And so typically analysis in time series requires a lot of data points. But for example, here we see that the trend is going up, as we know about global warming and so on, because we see a lot of number of data points, and so we see the trend going up. But if, for example, we take these 2 mini time series, you see these up and down trends in both of them, but it's the longest one that gives you the idea of the trend. So you need a lot of data points. But in single-cell RNA sequencing data, you're not allowed to have that many time points because of moral reasons, because of cost reasons, even though costs are going down recently, or also actual feasibility. So usually you have less time points. So let's try to be moral in a way, not pay too much money, and trying to find a robust alternative that works with less data points and also especially captures all the heterogeneity that is given by single cell data. So we present SHISMA, which has this objective, as I said, that is identifying these subnetworks that are significant given by a p-value. Cell-type specific, shape-driven, and we will see later what we mean by that, integrating also a priori knowledge through a protein-protein interaction network. The general outline is this one. The inputs is the time series single-cell data, the cell type identity of the time series, the genetic identity of the time series, and the protein-protein interaction network. And then there is this 3-step method, which starts with the primitive transformation, a score calculation, and then the actual subnetworks discovery. So let's go with the first one. Before, let me introduce a little bit about the data set. Since here we are talking about single-cell data and not really spatial, you cannot track the exact single cell over time, but you can track genes and populations. So it makes sense for the inputs to pivot to a gene, cell type, average expression over time. There are some tools that actually try to estimate and mimic the evolution of the single cell, but we're not going to talk about them today. So step 1, time series primitives needs to be introduced. Primitives are like basic operations or transformations that try to transform the time series dataset into an interpretable and simpler version of itself. We use for the step 1 the bag of receptive fields, which is a primitive that came out recently and it's getting popular and popular. And it starts with a preliminary step, which is based on an alphabet of symbols. Here, for example, it's really simple because it's high, medium, and low value, and it's pretty intuitive. And so we do this preliminary step of transformation. And then given a set of shapes that is essentially combinations of these symbols of the alphabet, we start with the actual transformation. And we see now what we mean by that. This is really simplistic version of the algorithm, but it's pretty explanatory. So we take a time series and we take a shape, in this case, for example, medium, medium, high, so MMH of length 3, and we create a certain window, the window of the shape, and we slide it through the time series. And we count essentially how many occurrences are happening on this time series. Here we have 1, we slide it again, 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 and again. We have the second one, and we put the number in the transform version. So it's a little bit more complicated than that because there are some parameters that you can control, but essentially it's that. And so you get this Z transformed matrix of the time series that essentially counts how well a certain time series is represented by the various shapes that you have. And so we go with the second step, which is the calculation of the score that is given by Shapley values. We fit a decision tree on this transformed version of the dataset to predict the genetic identity of the dataset. We use the decision tree because it's fast and accurate. We tried with also other ones that are tree-based and Shapley compatible, but this is the best one. And so you get this contribution score that is gene shape specific for this couple. And then you can also filter for a specific cell type that you want. The problem here is that you have a high number of data, a high number of genes and cells. So essentially you are prone to false positives. So you need to bound the family-wise error rate. So what we did is introduce a permutation testing by shuffling the genetic identity. And so after calculating the p-value of the permutation test, then of course you can keep the Shapley value if the p-value is significant. Otherwise, you just set it to 0 and you're good to go. We will see later the importance of this. And then we go with step 3, which is the actual subnetworks discovery. And for this, we took a version of the algorithm that is hierarchical hotnet from Reina et al. In 2018 that essentially works with random walk with restart. So you get this diffusion-like process along the network, the protein-protein induced network. So you put the scores in the original network, and then with this random walk, you can share this knowledge, this hotness, these scores to proximal nodes. And then with that, after the random walk is completed, then you get shared scores, and you can set a threshold to get the actual significant subnetworks out of the original one. And so you repeat this step for every shape, essentially, and you get a list of shape-specific, cell-type-specific significant subnetworks, or at least with a p-value. Then you have to check if it's significant for you or not. So in a nutshell, our contributions are these. We guarantee for multiple hypothesis testing, and then we also demonstrate why it's important. We develop this algorithm that is able to infer these subnetworks. And we extensively, of course, experiment on synthetic and real-world data that we'll see later. Regarding the synthetic experiments, I will only focus on this one for the sake of time, which is the multi-hypothesis testing. And so we created an alternate version, a fake version of SHISMA, which we call SHISMA asterisk, that has no permutation testing. So it skips the bounding of the family-wise error rate. And we give them a random data set, so time series that are taken from a random distribution with a varying number of sample sizes, a varying number of time points, and so on. And every time we give it to the SHISMA asterisk, the fake one, we have at least one significant subnetwork, so it's a false positive. because of course the original distribution is random and you cannot have from random stuff significant stuff. And instead SHISMA gives always no subnetworks. So we have 0 false positives. So informally, we can say that every return subnetwork that is given by SHISMA, it bounds the family-wise error rate given a certain confidence level alpha. So strong with this, we go on with the real-world experiments. This is the experimental setup. It's human donors, blood from human donors that was like immune primed, so to check what is the immune response in the samples. And it's 6 time points taken sample at day 0, 1, sorry, don't remember, 5, 9, 12, and 15. The data were normalized. And then for the results here, I will show you only the B cells because they have like a wide literature behind. And for the protein-protein interaction network, we use these 2 that are pretty popular. So this is essentially what you get in the end, a list of subnetworks that are specific for a cell type, in this case, B cells, with the p-value and the shape that is like simplified with these dots. So if we take a look, for example, at the first one, we see that these genes, the genes that are involved in this subnetwork, are protein synthesis related and also related to antibody production. And we see that the shape is actually like relevant to early proliferation. And so this makes sense with literature. Then if we go on and we take, for example, this other subnetwork, these genes have a clear signature of B cell maturation. So we are like in the middle part. Here the shape is more like up and down, and probably it's something related to fast interplay, but actually would need to be investigated more, especially in vitro. And then if we take a look at these 2, this is more related to protein folding and antigen presentation. So it's more like a late spike, as we see with the shape. So it's also consistent with literature. So let me conclude the presentation with like some final remarks. So we presented SHISMA, which is this algorithm able to infer these significant subnetworks, integrating also a priori knowledge. We demonstrated, as we saw, the importance of guaranteeing for multi-hypothesis testing in the time series scenario. So we experimented in synthetic and real-world data, finding coherent results. And in the future, we would like to try with other data. For example, we have the possibility to sequence breast cancer data over time. But we need money, so I applied to a grant, and hopefully I will get the money. Maybe they will tell me now because they told me 22nd of July, so should be maybe by tomorrow I will tell you if I can go on. So this is the conclusion. Let me just do some little bit of marketing. This is the QR code of my website. All the updates will be there because the paper is not out. But when it will be, if it will be there. You can connect via LinkedIn, Twitter, whatever. These are the people that are like part of this project. Especially Pierangela is here, so if you want to do like hard questions about the biology, she's the one. And then also Emilia Vigolo and Francesco Spinato. With this, I thank you, and I'm ready to take questions, if any.
Thank you very much. Questions?
Very interesting. I wonder how sensitive it is to dropouts, the way you normalize your data, and in general batch effects, especially if you have a time series and you would have quite a lot of batch effects, I would imagine.
It's a good suggestion. Actually, regarding the normalization, we actually haven't tried with different ways to normalize the data, if that's what you mean. But actually, we tried in another project with some master thesis student to try to normalize the data with different tools. and actually the difference regarding, for example, the cell type identity doesn't change much. So actually, this should be, it's like the technique, the method that is robust in principle. But regarding dropouts, probably totally makes sense to do a test about it. And I will certainly do that. So thank you for the suggestion.
Thank you so much for the wonderful talk. I was wondering, if you don't have time series data, could you use something like RNA velocity as a proxy to create those time points and use that?
Wonderful question. I actually thought about that in the beginning. But I think that, yeah, if you can create a time series that is like similar to this, then yeah, of course. The problem with, for example, RNA velocity trajectories and so on, is that you don't have like clear time points. So it's more like a flow. So actually, if you are able to select like exact time points, because as you saw, we need shapes, predetermined shapes. So if you are able to determine time points, then yeah. If you have like more like a flow, it's more complicated. But yeah, thank you.
Thank you so much.
Thank you.